I'll talk a little bit more about quorum sensing and some other ideas, but then I'll set aside some time. We'll go into the second exam and go through some of that, okay? I analyzed the stats for each question and I ended up adding 8 points to everybody's score, okay? So we'll go through that yet today. But first, let's get into a little bit more of quorum sensing. And I think we left off here with this idea of how we can even see bacteria in our gastrointestinal tract can be the quorum sensing process for a variety of reasons they're finding out. Now, with this particular example, it's the idea that one bacteria could potentially be producing a chemical signal that can actually mess with Vibrio's ability to make you sick. And so in order for Vibrio to do that, it has to do its own version of quorum sensing. And they're finding that other nearby bacteria can potentially disrupt that, which is definitely a good thing. Let's try an eye clicker before we get going too much further. Let's see what we can do. Whoops. That's not what I wanted. Which quorum sensing system involves the chemical signal binding with the regulatory protein? Are we done clicking in? Let's see if I can get it to show. Okay. So we have this idea of the quorum sensing signal itself binding with the regulatory protein. The regulatory protein's job is to regulate gene expression. So you have to think about the version that has the chemical signal diffusing into the cell. It has to in order to bind with the regulatory protein, and that's the gram-negative cell type, the gram-negatives. So this is where you have the gram-negatives all producing homoserine lactone, and then eventually the threshold level will get met and you'll get diffusion of the homoserine lactone back in to the cell, which binds with the regulatory protein, and then that complex will bind with the DNA and give gene expression. Okay. One more. Let's do one more. Let's see if I can get it to work here. Here, I don't know if I can do it this way. It's gonna freak out on me. So can you probably not read that? Let me get this out of the way. With gram-positive bacteria, which protein gets phosphorylated, which then gets the protein to interact with DNA and alter gene expression? I think that's everybody. So most of you said B, response regulator. That's correct. Okay, so this is the two-component signal transduction system where the sensor kinase interacts with the peptide pheromone, and then it will pass a phosphate off to the response regulator, and that binding of the phosphate to the response regulator is kind of waking up the response regulator and telling, hey, now it's your job. Do what you need to do, which is to bind to the DNA and alter genes. So some differences there between the typical gram-positive two-component system versus the gram-negative. So as you're studying this, make sure that you're looking at what's similar, what's different when you see gram-negative versus gram-positive. Questions on the basic process? All right, so we see that microbes, depending on who they are, have slightly different methods. Whoops, let me do it one at a time. So we're also finding that not just bacteria doing this. We can see that other organisms that often interact with bacteria can also play this game. And so there's been a lot of study with plants and their associated microbes, both the beneficial microbes as well as pathogens, and whether or not they can communicate with each other through this quorum sensing process. And the short answer is yes, they can. So plants can recognize quorum sensing signals that are coming from the bacteria, and they can recognize those signals and respond appropriately to them. And I'll give you some examples. They also know the reverse. Plants can actually make quorum sensing signals that the bacteria can respond to. AHL is a homoserine lactone. So most of it has been done with gram-negative bacteria and how gram-negatives interact with plants. They've done studies where plants can degrade quorum sensing signals between bacteria, so stop the communication and the behavior that might result. So that often that's when you think about pathogens trying to attack the plant. On the other hand, we also see that plants can stimulate quorum sensing, especially with beneficial bacteria that are hanging out with the plant. So the plants will encourage that quorum sensing process with those bacteria. And so they've done a lot of work with the beneficial bacteria and how plants are stimulating that. They've looked at pathogens, the negative effect as well. And probably these are the big plants that they've been studying: peas, rice, soybeans, and tomato, all really important in commercial crop production, which is why they tend to focus on these types of plants. So the idea here is, if we can better understand quorum sensing, and then we can start applying it to real world situations. In this case, it's to help plants thrive and produce things that we want them to produce, so we can end up harvesting them and eating them. And so here's some other examples. There's definitely the ability of the host, like I said, with the red algae example I gave you the other day, terrestrial plants as well, that they can negatively affect the pathogen. So they can mess with the quorum sensing process of pathogens trying to attack the plant or the red algae. Here's a kind of a interesting process where it's pretty sneaky, actually. So the plant host may actually stimulate quorum sensing response in plants. So this may sound counterintuitive, like why would a plant want to encourage a pathogen? What they do, it all depends on the timing. What they do is they will secrete out some quorum sensing signals that the pathogen can respond to. And what happens is the pathogen will be at low population levels. So let's say there's like 100 cells. And normally that's not enough for the pathogen to kick in the quorum sensing behavior. But if they pick up this signal, these quorum sensing signals coming from the plant, they get faked out. They're like, oh, there's all these quorum sensing chemicals. Oh my gosh, there's a bunch of us around here. We can go ahead and mount an attack. But there's really only 100 of them around. And it's actually these signals coming from the plant. And so it's like the plant is tricking the pathogen, getting the pathogen to reveal. What happens next is the pathogen says, oh, there must be a bunch of me around, so we can go ahead and attack. And so they start making virulence factors, proteins and enzymes. And that can actually kick in. Once those get released, the plant will respond to that and mount a defense. And so it's this really kind of tricky way for the plant to fake out the pathogen. Pathogen starts to kick in to producing these toxins. The bacteria or the plant picks up on it, goes back, and starts defending itself when there's only like 100 cells. Now, if the pathogen had done that when there was 1,000 or 10,000 cells, it would have been a totally different story. So when this was published, it was really quite stunning that the plant was essentially tricking out the pathogen and making the pathogen kind of reveal its hand before it was ready to be attacking. It was a successful way for the plant to defend itself and fight off the pathogen. And then just the opposite too, the plant can release quorum sensing signals, AI autoinducers, or quorum sensing signals to actually encourage beneficial microbes to get into their beneficial quorum sensing behaviors. So there's both ideas here that the plant can interact both with pathogens, trick them out, and with the beneficials to really encourage them to stay near the plant and engage in that quorum sensing behavior that's beneficial to the plant. So there's been lots of studies looking at this. I tend to, because I study plants, I'm more immersed in what's going on in the plant realm. We'll talk a little bit about humans too, but here you can see the title of a published paper, that plants are able to stimulate or deal with pathogens, either stimulate the good guys or deal with the pathogens. And then interkingdom signaling is just getting at the idea that you have two different kingdoms of life. You have kingdom plantae and then, bacteria, very unrelated organisms, and yet they're able to take part in this quorum sensing process. So this was something that was really kind of stunning when it first came out, that there's that level of communication between vastly distantly related organisms. And so now they're starting to realize that it's not just plants and their associated microbes. We also can see it with humans and their associated microbes. And so it's starting to, and animals, of course, other animals besides us. But probably the first work that was done was with pea plants. And they were looking at bacteria that clean out pea plants. And it's the bacteria fixing nitrogen for the plant. And they were trying to understand that partnership. And they realized that quorum sensing has to happen in order for this to occur. And so that's kind of where it all kind of broke open. People started studying it very intently after that. And then when the human microbiome studies came into effect, which was, 15, 16 years ago, I think now. I think it was back in 2005 when the government, primarily the National Institute for Health, gave out millions of dollars of grant funding for scientists to look at the normal population of microbes in the human and looking at how they interact with the human host. And here you're seeing what they're trying to draw here are some epithelial cells from the gastrointestinal tract. And they're trying to show how epithelial cells are actually releasing those AI2 quorum sensing chemicals that can be picked up and understood by some of the bacteria in your gastrointestinal tract. And so this is actually happening probably in you right now in your gastrointestinal. So this idea that you have, you know, a human and its associated microbes interacting in this way, again, it was kind of a big aha moment when this kind of research was starting to get published. And so they're really starting to ask, you know, how can we use this information to improve human health and other animals? Here's an example of one study that they did where, you know, if you take, I'm sure most of us in this room have taken antibiotics by now, and you probably, most of the antibiotics that humans take when you're sick, they're called broad spectrum antibiotics, which means they end up killing lots of microbes in you. They're not just targeted to the particular pathogen you're sick with. So it's not great to have to take those because it kills off a lot of your good microbes. And so here they're showing that idea of how antibiotics are disrupting the microbial population in these systems that they were studying. I can't remember if it was mice or mice that they were looking at. And so here they're showing that Firmicutes and Bacteroides are two big groups of bacteria that hang out in our intestinal tract. And what they're showing is the antibiotics preferentially knock out the AI2 to now you see less green cells. The Firmicutes have gone away. And they're saying that created a dysbiosis, which is pretty much an imbalance of microbes in you that is not ideal, negative thing. And so here what they're showing is that if they had probiotics taken of E. coli that was engineered to produce AI2, when that got into the gastrointestinal tract, it helped restore back close to the normal population for antibiotic exposure. And so they're looking at this idea of, you know, kind of enhancing with quorum sensing signals like that with this probiotic AI2 is able to help get that population back to normal. With the AI2 in with the probiotic, what they think happens is that it allows for basically in this case, it would allow the Firmicutes to come back into balance. And yes, the idea of, in this particular case, when it helps stimulate the growth of Firmicutes. And so we'll talk a lot more about Firmicutes and Bacteroides, but they're starting to mess around with this idea of, engineering probiotics that have quorum sensing signals under the right conditions could be beneficial for the host. Bonnie Bassler's work is also looking not just at bacteria, but she's also looking at, do bacteria and viruses interact in a quorum sensing mechanism? And so basically the short answer is yes, and which was kind of surprising considering virus particles. We don't really consider them to be living entities, but she was looking at bacteriophages, which are viruses that have bacteria. She was showing that they are using quorum sensing systems in order to interact with each other. And so, you know, the bacteria are trying to do it to keep the phage away from them, whereas the phage are trying to do it in order to successfully attack. And so it gets complicated. The more they look into quorum sensing, the more detailed and I guess complicated it can get. And so, this idea that there can be, these interactions between organisms where you have some kind of disruption going on in the quorum sensing process. Sometimes that could be very useful. And that's the idea of quorum quenching that I mentioned the other day. And so looking at this idea of warfare again, if you can mess with your enemy's ability to communicate with each other, you're probably in a better situation to win the war. And that, historically, that's held true back in the history of World War II and one in particular. They could mess with the enemy's communication system. That was a very successful strategy. And so that's what quorum quenching is trying to get across here. Here's one example where they're looking at endophytic bacteria and they were showing that you could get endophytic bacteria could mess with quorum sensing chromobaccterium, which can be pathogenic. And so this idea of, in this case, it's an endophyte, which is a bacteria that lives inside of a plant. In this case, they were studying cannabis. And so they're starting to look at these types of interactions, and if we can better understand it, then we could do more quorum quenching instead of antibiotic therapy or something like that. The idea of quorum quenching is pretty attractive because microbes really can't gain resistance to that, but they can antibiotics. So I just put this here to show you kind of behind the scenes what they would have to do in order to look at some of these ideas in real life. So here they're looking at getting your plants grown, you have to isolate the fungi, and you have to purify them. So and then start working with them to better understand them. You get their DNA analyzed, of course, and on and on and on to where you learn more about them and then start doing your extraction. So it's a step by step. This can take months or years, more typically years, but it can be done and is something that is being worked on right now. So this idea of quorum quenching within your lifetime, I think you will be given quorum quenching drugs. Eventually, when you're sick with some pathogen, that will be a route rather than just giving you a bunch of antibiotics that may not even work anymore anyways. And so this is becoming a big thing in the endophyte realm where they're looking at plants, the endophytes, the microbes that hang out in the plants, and trying to see if they can be used to help quench quorum sensing in different plant pathogens. But humans too, they've done some studies with humans. The epithelial cells, one of the studies I read was actually in the oral microbiome. So it was the epithelial cells, like the cheek cells and gum cells in the mouth. And they were looking at this idea of that there was quorum sensing going on by some oral microbes, and that the epithelial cells there were able to quench that signaling process. So they're starting to look more carefully. They're finding it in the mouth of humans. They're finding it in the intestinal tract of humans, that they can be releasing quorum sensing disruptors and do the quenching process. They're also finding just natural systems out there in the real world, soil systems. They found that there's different microbes in the soil that can do quorum quenching. So bacillus species can break down, cleaves right where the lactone ring structure is. And if you break down the chemical, that's very successful. It can't be used anymore as a signal. Soil streptomyces are doing the same thing. They're able to break down cycles. Would that mean that in the as well, could do it that way. Basically, the pathogen won't engage in attack unless they can determine, oh, there's enough of us to do this successfully. So if you can quench the signal. Basically, they say you just make it so that the quorum sensing signal the pathogen is putting out, if you can like degrade it or bind with it so that it can't do its job, then the pathogen's like, well, I guess there's not enough of us around because they're not getting that signaling process to kick in anyway. And so they're just sitting there going, yeah, I guess we can't attack yet, even though there may be some population level that's good, but because the quorum sensing chemical was messed with, somehow quenched in some way. And so again, they're not really, if you were to take this, it would kill the pathogen directly. The pathogen would still be in you, but what tends to happen is even if there's like 1,000 of these pathogens in you, technically, you know, if their quorum sensing is working, they'd be attacking you, but because you took a quenching chemical, they're not. But then your immune system can figure out eventually, we got to wipe these 1,000 pathogen cells out.
So that makes it so that in just like I guess
right. And that's what they're saying. You may still get some baseline response, but you, because they're not shifting into pathogenic behavior where they wouldn't be pumping out all these toxins, virulence factors to attack you, they wouldn't do that because they're getting tricked into thinking there's not enough of us around, so we can't do it yet, even though in reality there may be. So, and that's, you know, they can't really gain resistance to that. Like when you take an antibiotic, you know, they're very quick to gain resistance against that. They don't think that's going to happen with these quorum sensing quenchers. And so they're really excited about it, but you know, I don't think it's there yet. But I think within your lifetime, you'll be taking drugs that will pretty much be doing that. And so, and then you have to rely on your immune system to finish off. So, you know, they're finding it in natural organisms out in the soil or environment. In this case, they're finding it with host microbe interactions. So it's universal, not necessarily just with a host microbe interaction, just normal microbes out there living in a soil environment, for example. So, you know, if they are quenching it just normally, like these Bacillus and Streptomyces species hanging out in the soil, if they're doing that, the question, you know, well, why? And if you look at, go back to this, if you look at the homoserine lactone in this case, or the peptides, those signal molecules, they could be used for nutrition. Maybe that's why they're breaking them down. You know, so it could be something just kind of, they just want it for food. They're not really deliberately breaking it down to block quorum sensing. However, there is the other hypothesis that depending on the context of the situation, they could be deliberately doing this in order to quench the quorum sensing process. And so, you know, there's a lot of interesting studies being done to get at this a little bit better. And so, yeah, definitely they're looking for how do we use this information, because if you talk to any medical microbiologist, they're pretty concerned about antibiotic resistance, and they're trying to find other mechanisms besides giving patients antibiotics. And so looking at these drugs, they're calling them quorum quenching drugs or quorum sensing blocking drugs. And basically, yeah, you can just disrupt the signaling process, then those microbes won't engage in the behavior. And with pathogens, that's great. We don't want them to engage in pathogenic behavior. And so they're gaining ground on this. They've done some good work on it, but it's a little ways off yet. So speaking of gaining ground, this next image is looking at some of the chemicals they have discovered. So these are quorum quenching chemicals. And you see there's a variety of different chemicals. It's not like there's one or two. There's a lot of options there. And so they're working hard on this. And here again, they're trying to figure out, okay, if you know that compost, like human, can be released for quenching, how are they doing it? What are the methods? And so they say, well, maybe they're somehow modifying the chemical signal so it can't work anymore. It should. or maybe they're breaking it down, or maybe they're sequestering it somehow. So they still aren't, like how do we, they're trying to figure out natural quenching systems and then work with that for drugs, to make drugs that, and you're sick, you need to take a drug, and they don't want you to keep taking antibiotics. So it's lots of work to do, but it makes you wonder too, the other way, can bacteria that are in our bodies, what might they be doing to the host? Could they be impacting, could the quorum sensing process mess with our immune system? And that has been asked and looked at. And the short answer is yes. And so basically they're finding that, like Pseudomonas aeruginosa again, that it helps them evade our immune system if they can engage in quorum sensing behavior. So there's that type of interaction too, where they become so fully evolved with hosts that they use quorum sensing to mess with the immune system. So not good, but again, really important information. This particular study was done and they were looking at both the gastrointestinal tract and again in the oral microbiome. And so with a tooth root tip, they were actually investigating that. So, we have an incredibly sophisticated immune system, but microbes are using quorum sensing to deal with that. So that's not such a good idea, but it's happening. Okay. I think I'm going to stop there and go over the exam. So I don't know that now that I'm looking at my dumb computer here, I don't think it did the Zoom recording very well. So this might not be, you might hear the audio and visual. I don't know. Frustrating.